Okey, Assalamualaikum warahmatullahi wabarakatuh. Okey, harap-harap semua dah ada. Okey. Okey. Okey, selamat datang. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Selamat datang ke program e-revision 1.0 sesi yang kedua. Dan selamat berjumpa kembali lah. Sebelum ni kita berada pada program e-revision 1.0 sesi yang pertama. So sekarang hari ni adalah sesi yang kedua. Okay. So saya adalah Puan Hidayatul Ilah binti Ahmad Saad, merangkap sebagai moderator untuk program malam ini. So saya berharap para pelajar semua sihat dan cergas dan sudah bersedialah untuk mendengar perbincangan bagi soalan Biofortune set 2. Okey, semua kena bersedia dengan soalan tersebut. Okey. Program ini, perbincangan ini akan disampaikan oleh dua orang penceramah kita, di mana kedua-dua penceramah kita merupakan penceramah yang sangat hebat, pensyarah yang sangat hebat dan berpengalaman, dan juga merupakan expert dalam konten biologi. Jadi saya mohonlah para pelajar mengambil peluang ini untuk menyemak jawapan anda dan memahami setiap poin semasa perbincangan tersebut dijalankan. Okey. Saya difahamkan para pelajar berpeluang untuk merangkul hadiah yang lumayan jika terus setia mengikuti program pada malam ini. Okey. Jadi so ambil peluang ini untuk semua dapat menguatkan ataupun memantapkan lagi tahap pemahaman bagi setiap soalan yang akan dibincangkan sebentar lagi. Okey, baiklah, sebelum saya menjemput para penceramah untuk meneruskan perbincangan mereka, so terlebih dahulu saya ingin membaca biodata berkaitan kedua-dua penceramah kita pada malam ini. Okey, penceramah kita yang pertama adalah Puan Zaiti, Puan Nur Zaiti binti Bakri, di mana Puan Zaiti ni merupakan pensyarah yang berpengalaman mengajar di Kolej Matrikulasi Pulau Pinang selama 5 tahun, dan beliau juga merupakan pemeriksa kertas jawapan biologi, dan juga beliau adalah alumni program pelajar cemerlang PPC. Okay, seterusnya penceramah kita yang kedua adalah Puan Tamila Rasi, anak perempuan Raja Ram. Puan Tami ini merupakan pensyarah yang sangat berpengalaman. Beliau telah berkhidmat di Kolej Matrikulasi Kolej Matrikulasi Pulau Pinang selama 12 tahun. Okey, dan beliau juga merupakan penggubal soalan UPS dan juga merupakan pemeriksa kertas jawapan biologi. Okey. So baiklah. Tanpa kita membuang masa lagi, saya ingin menjemputlah penceramah yang pertama, iaitu Puan Zaiti, untuk memulakan ceramah beliau. Okey. Puan Zaiti, apa khabar?
Okay, Alhamdulillah. Okay, Assalamualaikum and good evening to everyone, to all the students and all the lecturers yang join Bengkel Pecutan 2.0 ni. So insyaAllah hopefully semua orang, tak kisahlah pelajar ke lecturer ke, we can stay until the last lah untuk membincangkan all 30 question in Biofortune set 2. Okay, so insyaAllah kita akan sama-sama mengupas setiap soalan dan kita clarify lah apa-apa part-part dalam biologi ni yang kita tak faham. Okay. Okay, so Assalamualaikum, Adham, okay, evening Afika. Okay, thank you Puan Syuhaiza, okay, sebab join. InsyaAllah semoga ilmu malam ni dapat dimanfaatkan oleh semua orang lah. Okay, Andai kata ada soalan along the way daripada sesiapa pun lah berkenaan tentang part yang kita bincangkan ke ataupun berkenaan tentang topik yang kita sedang bincangkan ke, feel free untuk tulis je soalan tu dekat ruangan chat lah dekat YouTube live. So don't worry, kalau saya tak sempat nak cater pun, kita ada lecturer biologi yang lain lah yang tengah standby untuk menjawab soalan-soalan yang ada. So saya rasa kita akan teruskan je lah kot dengan perbincangan kita untuk BioFortune MCQ set 2. Bersama saya, andai kata sesiapa yang saya ajar mungkin dah kenal lah kot. Saya Madam Nurzaiti binti Bakri. Tapi kalau siapa yang ini baru first time kita bersua, selamat berkenalan. So insyaAllah lah semester depan kalau ada rezeki mungkin kita akan berjumpa lah kan, face to face kan. Thank you Putri Nur Ain, Cahaya. Thank you. So saya start dengan soalan yang pertama. So hopefully semua orang boleh standby jugalah dengan kalau sesiapa yang dah ada hard copy untuk BioFortune MCQ set 2, boleh sediakanlah, boleh keluarkan hard copy tu untuk kita take note lah any part. So untuk soalan yang pertama, so hopefully semua orang ready. So untuk soalan yang pertama, if a DNA molecules contain 20 G. So what is G? 20 G is 20% of guanine. So we know that DNA molecules, dia adalah double stranded and it contains 4 different types of bases lah. Guanine, cytosine, adenine and thymine. So this question asks, if that DNA molecules contains 20% of guanine, so what would be the percentage of adenine? So there are multiple choices answer here. Either the answer could be 15 or 20, 25 or 30. So siapa-siapa yang rasa dia tahu jawapan dia, boleh put your answer in the comment. So kita tengok explanation dia. So from this diagram, as you can see, kita ada structure of nucleic acid of DNA. So we know that nucleic acid consists of double helix lah. Maksudnya dia adalah consists of 2 strand of polynucleotide. And this 2 strand actually is connected between their bases. So as you can see in the diagram, you can see that cytosine partner with guanine and adenine partner with thymine. So same goes to down here, guanine with cytosine, and also we have thymine and adenine. So this is fixed. Maksudnya partner mereka memang dah tetap. Adenine will always form 2 hydrogen bond with thymine, and cytosine will always form 3 hydrogen bonds with guanine. So this is fixed. So kena take note ni lah. So from the question, simple je. Bila soalan tu dia berikan dalam bentuk percentage, so we can assume that the total number of the nucleotide in the DNA is 100%. It depends on type of question. Kalau question tu dia letak dalam bentuk percentage, kita boleh assume dia sebagai 100% lah. Tapi kadang-kadang ada setengah soalan dia akan bagi total number of nucleotide. Tak kisahlah, mungkin dia akan bagi 200 ke, 300 ke, so depend on type of question. So for this one, we have in percentage, So it's stated that where guanine has 20%. So we know that guanine will always partner up with cytosine. Dia memang akan sentiasa buat hydrogen bond dengan cytosine. So logic thinking, if you have 20% of guanine means we are going to have 20% of cytosine too. So in total, we have 40% for both cytosine and guanine. So how to find out what is the percentage for adenine? Simple. The total number of nucleotide, tolakkan dengan the total number of cytosine and guanine, which is 40%. Then you are going to have 60%, and 60% refer to both adenine and thymine. So since adenine will make hydrogen with thymine, means they are going to have equal number ataupun equal amount of nucleotide lah, percentage dia akan sama. So simply 60 divided by 2, you are going to have 30%. So we are going to have 30% for adenine and also 30% for thymine. Okay, so let's look at the answer. So the answer is D. Okay, so congratulations to everyone. Okay, good. Afika, Atira, oh, okay, Fazlina. So I think everyone give answer D. Okay, correct. Good, good. So first question, still okay lah kan? Okay, so moving to the next question. So second question, which is not true about parenchyma cells? Kadang-kadang setengah student ni, bukan setengah student lah, kadang-kadang saya pun dulu kalau sebagai seorang student, bila nampak je tissue, cell ataupun anything that related to plant, dia dah macam, oh my god, okay. Tapi don't worry, kita akan tengok, okay, about parenchyma cells, okay. So parenchyma cell is one of the example under ground tissue, okay, for plant tissue. Okay, so plant tissue, parenchyma cell is under ground tissue. Okay, so we have to find between all this statement, yang mana satu yang salah lah, not true about parenchyma. So is it parenchyma is the most fleshy tissue of fruits? Okay, mostly composed of parenchyma cell. Adakah parenchyma cell have thicker primary cell walls than collenchyma? So adakah photosynthesis occurs within the chloroplast of parenchyma cell of the leaf, or most parenchyma cells retain the ability to divide and differentiate into other types of plant cells? So basically, there are three statements that is correct about about parenchyma cell, tapi ada satu akan salah lah. Okay, so boleh, semua orang dah start dah untuk bagi pilihan jawapan masing-masing. Okay, so kita tengok dia punya explanation. So simple. Parenchyma ni, okay, when you look at the characteristics, so it must be a living cells, okay, and the shape is must be isodiametric. So isodiametric ataupun elongated lah. So what is mean by isodiametric? Dia tak ada bucu, okay, dia bulat macam ni, okay. And kalau elongated ni, macam sebelah ni lah, okay, ni contoh elongated, okay. And what else? So parenchyma has a very thin primary walls that made up of cellulose, hemicellulose, and pectin, and there is no secondary cell wall for parenchyma. Okay, and then it has a nucleus that push to periphery, and also it has a very large permanent vacuole, and also it has a very large intercellular A space. As you can see here in the diagram, kita nampak dekat di antara sel-sel parenchyma tu kita ada banyak. Kita boleh nampak ada A space, okay, intercellular A space, okay. So here we have the function for parenchyma. So basically it has a basic packing tissue, and also it has a tergid cells that give support to herbaceous plant, and it's also important for store food such as starch, and also for gases exchange, and also parenchyma is important for photosynthesis, okay. So if you refer to your notes, okay, parenchyma ni it can be modified into different structure, okay. Salah satu dia adalah epidermis. Of course lah, we know that epidermis to provide protection, okay, and satu lagi adalah mesophyll. So you have to take note, mesophyll cells is very important for photosynthesis. Why? Because it contain chloroplast, okay, ada banyak chloroplast dekat dalam mesophyll cell, okay, which is part of sejenis parenchyma cell, okay, untuk buat photosynthesis, okay. So all this is the characteristic of parenchyma. Tapi saya saja je nak sentuh sikit berkenaan ketiga-tiga jenis ground tissue yang ada dekat dalam, ketiga-tiga jenis ground tissue ni. Selalu student keliru, macam mana kita nak differentiate parenchyma, collenchyma, sclerenchyma. So for me, dululah tips saya nak belajar, nak kena hafal. Okay, saya akan susun mereka ni ikut susunan begini. Parenchyma, collenchyma, and sclerenchyma. And I'm going to imagine them sebagai satu structure di mana parenchyma paling lembut, collenchyma ni tengah-tengah, and sclerenchyma ni memang paling kasar dan paling keras. So if you look at to the characteristic, okay, kita boleh kaitkan dengan tekstur yang saya sebut tadi tu, lembut, sederhana dan paling keras. Okay, contohnya untuk parenchyma. Okay, parenchyma dia mesti living cells. Okay, Dan collenchyma pun juga dia adalah living cell. Contohlah kan kita manusia orang kata kan, kalau hati dia mati, kita akan bayangkan macam dia orang yang keras lah hati dia kan. Tapi kalau orang yang hati dia hidup, maksudnya dia orang yang tenang, lembut. Okay, so bayangkan juga mereka yang living cell ni, tekstur dia masih lagi lembut lah. So parenchyma ni lembut lagi, collenchyma ni kita bolehlah kategorikan dia sebagai lembut lagi. Tapi untuk sclerenchyma, okay, memang tak langsung lah sebab dia adalah non-living cell. And when we look at to the shape, okay, shape of the parenchyma, isodiametric. Maksudnya dia bulat je. Kita kalau bayangkan benda bulat ni, kita memang akan bayangkan benda yang smooth je. Okay, bayangkan batu, kalau batu sungai tu, okay, dia dalam bentuk yang bulat. Kita nampak dia licin. Okay, tapi berbeza pula dengan collenchyma, yang dia punya shape is polygonal. Polygonal tu maksudnya dia ada banyak bucu. If you look at to the diagram, kita nampak dia dah ada bucu-bucu macam ni. Ni bayangkan batu kerikil lah. Batu-batu tepi jalan tu kan dia bucu-bucu, so dia agak kasar. Sclerenchyma dia ada different and variable shape lah. Dia tak ada specific shape. Okay. So dan seterusnya kalau tengok parenchyma. Parenchyma ni dia memang takkan ada, dia akan ada very thin primary cell walls. Okay, dia tak ada secondary cell wall langsung. So bayangkan cell wall dia pun nipis je. Dia taklah keras. Okay, manakala untuk collenchyma dia punya primary cell wall uneven thickness dan dia masih lagi tiada secondary cell wall. Tapi bila kita masuk kat sclerenchyma, dia dah lah ada primary cell wall. Lepas tu dia ada thick secondary cell wall. So kita boleh bayangkan bila dia punya secondary cell wall tu thick, dia akan jadi sangat kasar dan keras. Okay, dan yang terakhir, karakteristik yang saya boleh kaitkan dengan dia punya structure, dia punya texture dia adalah untuk parenchyma dia adalah loosely packed dan dia ada large intercellular space, manakala collenchyma closely packed tapi dia ada very small ataupun kadang-kadang tak ada langsung intercellular space. Manakala sclerenchyma very closely packed and confirm tak ada intercellular space. Okay, so kita bayangkan macam sponge lah. Sponge kan dia ada banyak lubang-lubang, so dia lembut. Okay, so that is how I imagine the structure for each types of ground tissue, parenchyma, collenchyma and sclerenchyma. Okay, so back to the question just now, which statement is not true? So we can see that most of the answer, ada yang tak sure, Haja cakap B tapi dia tak sure ke tak. Okay, don't worry Hanisha, mari kita tengok jawapan dia. Okay, so jawapan dia adalah B. Okay, so not true about parenchyma cell adalah it has a thicker primary walls than collenchyma. So of course salah lah. Okay, tak ada benda yang thick lah pasal pasal parenchyma ni. Okay, semuanya tin-tin sahaja. Okay. Okay, thank you for all the answers. Okay, saya tak boleh sebutkan nama tapi thank you Yusri, Christina, okay, Syahira, Aidul Mashita, okay, for all the correct answer. So moving to the next question. Number 3. So figure 1 shows a pair of centrioles. Okay, so you have to choose the best answer to describe their function. So apa benda centriole ni? Harap-harap kenal lah organelle ni. Okay, harap orang kata centriole ni dia taklah se-famous mitochondria, Golgi body. Okay, so ada jugalah yang tak kenal centriole ni. So which one is the best answer? Okay, is it centriole is for breaking down of the nuclear membrane, or maybe it helping in the pairing of homologous chromosome, or maybe it organizing of the microtubules to form spindle fiber, ataupun dia akan holding together the sister chromatid of a chromosome. Okay, so let's look at the characteristic of centriole. So here are the characteristic of centriole. As you can see here, okay, the structure of the centriole, it must be hollow cylinders. So as you can see structure dia kita nampak, okay, dia ada banyak bulat-bulat ni, and bulat-bulat ni sebenarnya bentuk dia hollow, dalam dia kosong. Okay, so it's a hollow cylinder and it always presents in pairs. Okay, dia mesti lagi bentuk dia dalam pair lah, satu yang ni, satu lagi yang bentuk macam ni, dan dia mesti dalam kedudukan right angle 90 degree. Okay, and dia akan located dekat centrosome. Apa benda centrosome ni? Okay, centrosome ni adalah clear area yang ada centrioles dalam dia. Okay, so you just imagine kalau kita tengok dalam gambar sebelah kanan ni, okay, so this area, okay, we call it as centrosome. Okay, so what we have inside centrosome, kita ada centrioles, pair of centrioles, okay, dan juga kita ada ester. So what is ester? Ester ni dia adalah radiate from the centriole. So yang bentuk macam radiation keluar ni, which consists of microtubules ni, itu yang kita panggil sebagai ester. Okay, so centrosome ataupun centrioles, okay, is it's responsible to microtubule organizing center lah. Okay, di situlah microtubules akan grow out. Okay, bila masa kita jumpa microtubules ni, kita akan jumpa microtubules during cell division. Okay, so during cell division, we know that microtubules is very important to produce, to form spindle fiber. Okay, so dekat sinilah spindle fiber atau microtubule tu originate lah, which is from centrosome ataupun centrioles. Okay, so what will happen during cell division? A pair of centriole ni, they are going to replicate, and then each pair will migrate to the opposite cells, and later on it going to form spindle fiber. Okay, tak sempat saya tanya lagi. Semua orang dah berpusu-pusu bagi jawapan masing-masing. Semua orang pilih C ni. Okay. Ada juga yang mewakili kawan. Rafi kata C, Isha kata C. Kawan tu mungkin tak ada device kot kan. Kita share device sama-sama. Okay, mari kita tengok jawapan dia. Yes. So the answer is C, organizing of the microtubules to form spindle fibers. Okay, so selama ni kalau siapa yang tak berapa kenal sangat dengan centrioles ni, so sekarang kita dah kenal. Okay, so moving to the next question, number 4. So the following events, okay, occurs in both, ni take note in both prophase 1 of meiosis 1 lah, of course dan juga and prophase of mitosis, okay, except, so you kena cari yang terkecuali. Okay, yang bukan. Okay, maksudnya, okay, you have to find the differences lah between prophase 1 of meiosis and also prophase of mitosis. Okay, so yang mana satu berbeza bagi mereka? Adakah formation of spindle fiber, nuclear envelope disappear, tetrads form, okay, ataupun chromosome condense. Okay, so mari kita tengok what are the events that occur during prophase 1 of meiosis and prophase of mitosis. Okay, so in the diagram here, okay, I put two different events lah, maksudnya two different phase. The first diagram is about what happened during prophase of mitosis, and the second one is what happened during prophase 1 of meiosis. Okay, kalau saya dalam kelas selalu, saya memang akan bagi student saya untuk hafal numbers of points. Okay, contohnya in mitosis kita ada 4 phases, prophase, metaphase, anaphase and telophase. So you have to remember 4, 1, 2, 4. Macam tu lah. So tapi tak adalah nak cerita semua kan. So saya explain dulu about prophase. So what are the 4 points about prophase that you have to take note? I always ask my student to remember, during prophase, take note, there is always something will be formed, there is always something will disappear, there is always something will migrate or move, and there is always something coil and shorten. So the question now, who do all these things? Okay, so siapa yang buat apa? Okay, so kita tengok yang pertama, okay, form. So siapa yang akan form during prophase? Spindle fiber will form from the centrosome. Okay, and next, who's going to disappear? Nuclear envelope and nucleolus will disappear. And third, who's going to migrate? Centrosome ataupun centrioles yang akan migrate or move to the opposite poles. And the last one, who's going to coil, shorten and condense and thicken, that's all is chromatin. Okay, so there are 4 points that you have to remember in prophase. Okay, dan bila you dah hafal benda yang dekat dalam prophase ni, tak ada masalah kalau you nak pergi kepada prophase 1 of meiosis 1. Sebab kenapa? You just have to repeat back all the 4 points in prophase of mitosis, and you just have to add another 2 points only. Okay, apa 2 points tu? Iaitu synapsis and crossing over sahaja. So you dah boleh jimat dah. Okay, you dah boleh jimat space dalam kepala kita ni, tak perlu nak hafal banyak-banyak. Sebab apa yang berlaku during prophase 1, okay, 4 things that occur during prophase 1 is same as what happened during in prophase of mitosis. Okay, so sama je konsep dia, di mana microtubules akan form, okay, nucleolus and nuclear envelope akan disappear, centrosome will migrate to the opposite poles, and also the chromatin will condense, okay, and shorten and thicken and everything lah. Okay, kita tambah satu lagi 2 is going to undergo synapsis. So what is synapsis? Synapsis ni adalah apabila a pair of homologous chromosome, okay, sit together. Diorang duduk sama-sama. Dulu sebelum cell division, dia pun ni berterabur. Dia pun suka hatilah nak duduk mana-mana kat dalam nukleus tu. Tapi bila nak berlaku cell division, masing-masing akan cari partner masing-masing. Okay, so that formation, we call it as synapsis. So of course it's going to produce, it's going to form a pair of homologous chromosome ataupun kita panggil dia tetrads ataupun kita panggil dia pun bivalent. All, you can use any lah. Okay, so itu yang pertama. Lepas dia dah pairing, dah buat synapsis, dia nak buat apa seterusnya? Dia akan buat crossing over. So crossing over tu di mana? Okay, dia akan exchange genetik lah. Kalau kita nampak dalam gambar ni, okay, we can see that the homologous chromosome ni dia undergo crossing over. Dia silang tangan. Okay, dia silang tangan. Macam contoh kalau kita tengok couple-couple baru kahwin kan, lepas diorang dah partner up, diorang kahwin, kalau jalan ni asyik silang tangan je. Yang macam tu lah. Okay, itulah crossing over. Okay, so the point where the crossing over occur, we call it as chiasmata. Okay, ataupun chiasma. Okay, so kalau kita tengok antara prophase of mitosis dengan prophase 1 of meiosis, we can see that we have 4 points that same, but we have only 2 points yang berbeza kat sini. Okay, so dalam soalan tadi, kita kena cari yang ni lah. Okay, so kita kena cari yang mana satu dekat dalam pilihan jawapan tadi, okay, yang tak sama, yang bukan di antara 4 points ni, tapi dia di antara synapsis ataupun crossing over. Okay, mari kita tengok jawapan dia. So obviously jawapan dia accept adalah tetrad forms. Okay, dia takkan buat synapsis. Okay, so apa beza antara prophase 1 and prophase of mitosis adalah tetrads form. Tetrads will only form during prophase 1 of meiosis. Okay, di mana pairing of homologous chromosome. Okay. Okay, good with all the jawapan. Setakat ni semua orang perform InsyaAllah for flat semua orang ni bio ni. Semua, so setakat soalan nombor 4 ni tak ada lagi yang bagi soalan salah ni. Okay, so okay Jasmine, yang tu PM to P nanti kalau nak slide ni. Okay, so next question. Number 5. Okay, in a monohybrid cross, the genotypic and phenotypic ratios for the F2 generation are both 1:1. Okay, sini keyword dia tolong take note. Apa keyword dia? Yang pertama dia kata dia adalah monohybrid cross. So bila cakap monohybrid cross, dia boleh datang daripada Mendel's, okay? Dia boleh datang daripada salah satu deviation yang kita belajar. Deviation ni ada banyak ni monohybrid. Okay, incomplete, codominant, multiple alleles ada banyak. So apa yang berlaku pula dia kata dekat F2 generation dia nanti ratio untuk genotypic and phenotypic dia 1:1. Okay, so what does the ratio indicate? So adakah statement di atas merujuk kepada incomplete? Okay, adakah dia merujuk kepada dominant and recessive? Adakah dia merujuk kepada the presence of lethal gene? Okay, ataupun the presence of linked gene. Kadang-kadang kita bila jumpa soalan macam ni, kalau ada pilihan jawapan yang kita rasa macam kita tak pernah dengar pun benda tu kan. Kita rasa macam memang tak pernah belajar lah. Awak boleh potong siap-siap. Contohnya the presence of lethal gene lah, kita memang tak pernah sentuh kan. Okay, so kita ada 3 pilihan jawapan kat sini. So mari kita tengok, okay, satu-satu. So saya akan mulakan dengan linked gene dulu. So apa benda linked gene ni? Okay, linked gene ni dia wajib dia mesti ada kaitan dengan test cross. So take note. Selalu kalau kita nak tengok dia adalah linked gene ataupun tak, dia punya keyword dalam statement tu kadang-kadang dia akan sebut test cross. Ataupun dia akan sebut juga perkataan crossing over ataupun without crossing over. So kalau dia adalah linked gene with crossing over, okay, dia tidak akan menghasilkan ratio 1:1:1 ataupun 1:1. So what is the result? Okay, for the ratio adalah kita akan dapat 2 phenotypes are larger, which is come from the parental phenotypes, and kita akan nampak 2 other phenotypes are smaller, which is show the recombinant phenotype. So contoh dia nanti kalau you all dapat soalan, contoh dia ada banyak macam ni. Nampak ni? Kalau kita nampak the number, we can see the big difference between the numbers. We can see that 2 phenotype ada nombor yang sangat besar sampai 900. Okay, and ada dua number yang nombor dia agak kecil. Okay, hanya 185 dan 206. And kalau kita tengok yang nombor besar ni adalah parental phenotypes dan yang kecil ni adalah recombinant phenotype, confirm dia adalah linked gene with crossing over. Okay, kalau dia without crossing over, dia akan bagi ratio 1:1 lah. Tapi 1:1 ni dia akan tunjuk parental sahaja. Okay, no recombinant at all. Okay, itu adalah without crossing over. Okay, so itu adalah untuk linked gene. Seterusnya, okay, incomplete dominant. So bila kita cakap pasal incomplete dominant, the only and always example yang kita akan selalu guna adalah snapdragon. Okay, memang kita akan guna snapdragon. Contoh kita tengok genetic diagram yang diberikan kat sini. Okay, kita ada pure breeding between the snapdragon red dan juga white. Okay, so mak bapaknya satu warna merah, satu warna putih. Tapi tiba-tiba anak yang dihasilkan berwarna pink. Okay, so salah satu ciri juga kita nak kenal dia adalah incomplete dominant. Dia akan menghasilkan new phenotype. Okay, dia akan menghasilkan new phenotype. Maksudnya phenotype yang terhasil ni tak macam the parents. Not red, not white, tapi pink. Okay, lepas tu bila dia continue dengan dia continue dengan self-cross untuk mendapatkan F2 generation, kita nampak untuk genotypic ratio dia akan dapat 1 homozygous dominant R, okay, 2 heterozygous R and W, and juga 1 homozygous recessive for W. So kita nampak ratio dia 1, 2, 1. Okay, kita tengok ni dari 1 je, yang ni ada 2, okay, and yang ni ada 1. Okay, and when we look at to the phenotypic ratio pun kita akan dapat 1 red, 2 pink, and 1 white. Okay, so obviously sekarang ni saya rasa semua orang dah boleh dapat jawapan dia. Okay, so masing-masing jawapan dia adalah A. Okay, good. So the answer is incomplete dominance. Okay, so next, moving to the next question. So question number 6, short horn in cows, okay, is dominant to long horn, okay, while black coat is dominant to brown coat. So cows that are heterozygous, so take note dia punya soalan, okay, the cows is heterozygous for both characteristic. Okay, dia buat test cross. Okay, so test cross ni dia akan wajiblah crosskan dengan homozygous recessive. Okay, and dia akan produce berapa banyak? Dia akan produce 100 offspring. So soalan dia, how many offspring would have short, okay, horn and black coat. Okay, so kita akan start, okay, buat dia punya genetic diagram dahulu. Okay, so first step memang kena buat genetic diagram dulu. Melainkan kalau awak ni memang hebat yang boleh teka je lah, nampak soalan je dah boleh faham, dah boleh bayangkan genetic diagram tu dalam kepala, then it's okay. Tapi kalau tak boleh nak bayangkan, lukis dulu, conteng dulu untuk lukis genetic diagram. Kita nak buat dulu genetic diagram untuk kita dapatkan ratio dia supaya baru kita boleh kira lah calculation dia. So kita based on to the question, the test cross is between heterozygous for both characteristic. So S besar, S kecil, B besar, B kecil lah. And kita akan crosskan dengan homozygous recessive, wajib. Bila kita cakap test cross, it must cross with homozygous recessive. Okay. So for the heterozygous individuals, okay, the gamete produced, we have 4 types of gamete. Okay, we have both dominant allele, we have a combination of dominant and recessive allele, and also we have both recessive allele. Okay, untuk yang homozygous recessive memang ada satu jenis sahaja. Okay, S kecil, B kecil. And then you cross all the possible gamete with S kecil, B kecil tadi ni. Okay, so you are going to have the F1 generation with short black, short brown, long black, and long brown. So the ratio that we are going to have is 1, 1, 1, 1. Okay, so soalan tu dia nak apa? Soalan tu nak how many is short and black. Okay, betul ke dia nak short and black? Yes, dia nak short and black. So bila tengok short and black, kita tengok ratio dia adalah 1. Okay, so 1 1/4 adalah ratio bagi short black. Okay, so lepas kita dah nampak ratio dia 1/4, kita boleh buat dalam calculation. Total of offspring adalah 100, so 1/4 times with 100, then you are going to have 25. Okay, so the answer for shorthorn and black coat is 25. Okay. Okay, good. Okay, semua orang pun jawab D. Okay, so congratulations. Okay, thank you siapa yang tulis jawapan awak dekat komen ni. Okay. Okay, so moving to the next question. Kalau ada soalan, okay, boleh juga tulis dekat chat tu. Okay, tak semestinya jawapan je awak boleh berikan. Okay, kod-kod lah ada yang tak faham ke. Okay, in my explanation, okay, boleh tanya dekat dalam chat. Okay, so moving to the next question. Number 7. So you have to select the possible term used to describe the phenomenon when a red flower, okay, kita ada fenomena di mana kita ada red flowered snapdragon is crossed with white flower snapdragon. Ni kalau student yang dia suka tajuk ni, dia memang hafal, dia memang faham tajuk ni, dia nampak aja snapdragon ni, dia memang terus tahu jawapan dia apa. Okay, dia tak perlu nak fikir-fikir lagi dah. Okay, tapi siapa yang macam tak sure, okay, snapdragon ni and produce pink flower progeny pula, okay, mari kita tengoklah pilihan jawapan dia, adakah dia epistasis, codominant, complete dominant ataupun incomplete dominant. Okay, saya akan explain epistasis, codominant dengan incomplete je, sebab complete dominant ni dia refer kepada normal dominant allele lah. Okay. So, epistasis. Okay, these things korang tak belajar Okay, it's not in our syllabus. Okay, cuma it's just a new knowledge lah untuk tahu. So, apa maksud epistasis ni? Epistasis ni adalah apabila phenotypic tu, okay, phenotypic expression of a gene, okay, at one locus, dia akan alters, okay, gene yang ada dekat second locus. Contoh dia adalah color on a retriever. Okay, kalau siapa yang kenal retriever, a types of dog, okay, dia punya gene for the color of this retriever adalah B, tapi it will be affected by E. Okay, kalau kita tengok kat sini, okay, kalau dia adalah kalau allele E dia ada sama ada homozygous dominant ataupun heterozygous, dia takkan memberi effect. Okay, there is no effect on the color of the retriever. So kalau dia homozygous recessive, dia akan color brown. Kalau dia ada dominant, okay, dia ada dominant allele, dia akan jadi black. Tapi kalau kes kat atas ni, okay, walaupun you adalah homozygous recessive ataupun you adalah homozygous dominant ataupun heterozygous, the color will always yellow. Kenapa? Sebab EE ni dia akan ambil kuasa tu. Macam tu lah. So EE ni dia akan tutup, okay, karakteristik yang ada pada gene B tu. Okay, so inilah maksud epistasis. Okay, the expression of a gene is affected by other gene. Okay. So next, codominant kita dah belajar. Okay, maksud codominant tu adalah apabila both allele are dominant, so they are going to be fully expressed. So the example that we're going to use is MN black group. Okay, so salah satu clue dia pun nak tahu codominance ni adalah the pure breeding cross akan menghasilkan new phenotype. So apa maksud dia? If you look at to this diagram, okay, kita ada pure breeding M black group dan juga N black group. So the result is you are going to have new phenotype which is M N black group. Okay, so dia tak sama dengan dia punya parents. Okay, so it's going to produce new phenotypes. Okay, and the ratio pun adalah 1, 2, 1. Okay, so that is for codominance. Moving to the last one which is incomplete dominance. So yang ni pun kita dah tengok juga tadi. Okay, sebab tu saya cakap kalau student yang alert dengan soalan tu, kalau dia nampak snapdragon, dia dah memang tahulah snapdragon is the only example for incomplete dominant. Di mana, okay, the cross of the pure breeding, red and white, it will produce new phenotype which is pink color. Okay, So jawapan dia D, saya jawab D, Sakina cakap D. Okay, so D madam. D, ya baik. Ya, jawapan dia adalah incomplete dominant. Okay, tak menyabar dia nak tunggu jawapan daripada saya kan. Betul, tapi kita kena explain kot ada kawan-kawan awak yang tak faham juga kan. Okay, so jawapan dia adalah incomplete dominant. Okay, so next question number 8. Okay, the progeny of F1, okay, from a cross between, okay, between a true breed, okay, dominant round pea plant, okay, dominant round seed pea plant dengan wrinkle seed pea plant, okay, were self-pollinated. So maksud self-pollinated ni adalah, dia adalah buat self-cross lah. Okay, dia buat self-cross. So dia punya soalan, you have to predict result in F2 generation. So kalau kita tengok kat sini, this is the type of monohybrid sebab dia hanya bercakap pasal satu jenis karakteristik sahaja, iaitu the shape of the pea plant, sama ada round ataupun wrinkle. Okay, so dia nak jawapan dalam F2 generation. So kita kena start daripada true breeding untuk dapat F1 generation. Lepas tu kita akan sambung dengan self-cross untuk kita dapat F2 generation. Okay, so sama juga, siapa yang tak boleh nak bayangkan genetic diagram dalam kepala, lukislah. Okay, so ini cara lukis dia. Okay, so tadi saya cakap kita dah detect dia adalah monohybrid self-cross. So dia adalah cross di antara round seed, okay, dan juga wrinkle seed. Okay, when we talk about true breeding, it will always cross between homozygous dominant and homozygous recessive. Okay, so kita start dengan kita punya lukisan genetic diagram. Kita start dengan parent dia, okay, homozygous dominant dan juga homozygous recessive. And then kita akan dapat gamet dia, satu dominant R, satu recessive R lah. Okay, and F1 generation kita akan dapat heterozygous. Okay, and daripada F1 ni kita continue, okay, untuk buat self-cross. Okay, so kalau kita dekat self-cross ni pula, okay, F1 ni kita akan crosskan dengan F1, di mana heterozygous crosskan dengan heterozygous. So gamet, both of them will produce satu gamet with dominant R, satu lagi gamet with recessive R. So when you do all the cross, so you are going to have this, where one is homozygous dominant, two heterozygous, and one homozygous recessive. So we have three with round shape, satu lagi one with wrinkle. So soalan tu tanya tadi, predict the result in F2 generation. So F2 generation kita akan dapat 3/4 with round shape and 1/4 dengan wrinkle shape. Okay, so the answer is D. Okay, Alhamdulillah semua orang memang jawapan D juga. Ya, terima kasih kerana turut bersetuju dengan saya ya Rodia. Okay. Okay, oh beli dekat Shopee jawapan, hebatnya. Okay, macam-macam korang ni kan. Okay, so baguslah, jadi korang masih lagi alert. Okay, so jawapan dia adalah 1/4 of the seed are wrinkle. Okay, so that is the best answer. So moving to the next question. Okay, number 9. Okay, so an allele Y which control the yellow coat color of mice is dominant over the allele Y kecil which control the grey coat color. So dalam population, okay, so we are in genetic population now. In a population, the total number of the mice adalah 2,500. Okay, daripada 2,500, 400 are grey. So the question asks you to find the frequency of allele Y besar, iaitu dominant allele. Okay, so when you have to find frequency of dominant allele means the question asks you to find P. Okay, you have to find what is P. Okay, so let's look at the calculation. Okay, so we have a population. So this is a population. Okay, so inside the population, total number is 2,500 and 400 is grey. Of course lah, bila kita tolak, the rest which is 2,100 adalah yellow color. So grey dia adalah recessive. So confirm kita tahu genotype dia Y kecil. Tapi untuk yellow, take note it can be homozygous dominant, it can be heterozygous. So the question asks you to find what is P. Okay, so the tips is please always start with Q squared. Kalau soalan tu dia bagi dia bagi nilai recessive phenotype macam soalan ni which is 400, that's good. Tapi kalau dia tak bagi, tolak. Cari juga the number of recessive phenotype because it's very good to always start with Q squared. Okay, iaitu sama ada dia mewakili recessive phenotype ataupun homozygous recessive genotype. Okay, so always start with Q. The frequency of the homozygous recessive genotype Q squared, okay, equal to 0.16, which is 400 divided by 2,500. So the answer is 0.16. So from Q squared, okay, you can find the frequency of the recessive allele, which is Q square root 0.16. You are going to have 0.4. So when we have the answer Q, then you can proceed with the formula P plus Q equal to 1 to find P, okay, which is the frequency of dominant allele. So the answer that you are going to have is 0.6. Okay, so jawapan untuk 0.6 adalah B. Okay, nampaknya Ilya Khairani je ke yang sempat menjawab soalan ni? Dia je yang macam baru bagi tahu jawapan dia B. Ke yang lain tu jawab C, 0.4? Okay, kenapa Hajar jawab B? Kenapa? Jawapan dia bukan B ke? Okay, so the frequency of allele Y besar adalah 0.6. Okay, so jawapan adalah 0.6. Okay, so kita teruskan dengan soalan nombor 10. Soalan nombor 10, non-disjunction. Okay, kita masuk dalam tajuk mutation. So non-disjunction of sex chromosome may occur during meiosis 1 of spermatogenesis. Okay, so which of the following are the possible genotypes of the zygote if the normal ovum, okay, so bila kita ada normal ovum, so normal ovum adalah haploid, okay, fertilize dengan these two types of sperm. Maksudnya types of sperm yang abnormal lah, okay, yang abnormal because of the non-disjunction that occur during spermatogenesis. So abnormal ni it can produce either abnormal N plus 1 ataupun dia boleh menghasilkan abnormal N minus 1. Okay, so inilah dua jenis abnormal sperm. Okay, so kita kena lukis dulu. Okay, mari kita cek jawapan dia. Okay, so case yang berlaku dekat sini adalah non-disjunction during meiosis 1 of spermatogenesis dan dia akan produce two abnormal gamete. Betul ke? Betul ke dia akan produce two abnormal gamete? So kita kena lukis baru kita nampak. So this is the germ cell. Okay, primordial germ cell lah, di mana we have 44 plus XY. Apa maksud 44 ni? 44 ni refer kepada kromosom number 1 until number 22. Okay, so kita deploy kan? So kita ada 44 kromosom. And plus XY ni kita nak refer kepada kromosom number 23, which is XY for spermatogenesis. And the problem occur here is non-disjunction during meiosis 1, di mana if non-disjunction occur during meiosis 1, the homologous chromosome, okay, fail to separate. Okay, homologous chromosome fail to separate. So bila dia fail to separate, kita tengok dia akan end up dalam satu sel kedua-dua X dan Y ada. Dan satu lagi sel ni kosong. Kosong ni bukan langsung tak ada kromosom. Okay, dia ada kromosom yang lain tu, 1 hingga 22, tapi kromosom nombor 23 je yang kosong. Manakala sel ni sepatutnya dia ada satu je, tapi dia terlebih ada kedua-dua sel iaitu XY. Okay, and then during meiosis 2 normal, okay, kira dia berlaku disjunction, okay, dia separate. So maka awak bayangkan kromosom ni dia akan align at metaphase plate, lepas tu dia akan separate. So bila dia separate, okay, kat sini akan dapat satu X dan satu Y. Sama juga dekat sini akan dapat satu X, satu Y. So dia akan end up dalam sel masing-masing lah. Okay, so dekat sini dia akan menghasilkan abnormal gamete N plus 1, di mana dia menghasilkan 22 plus XY. Sama juga dengan yang ni. Manakala yang ni dia akan menghasilkan abnormal gamete N-1, di mana dia dalamnya tiada kromosom number 23. So, bila dia fertilize dengan normal ovum which is haploid, okay, dia ada bawa satu X sahaja, okay, yang ni pun normal, dia ada bawa satu X sahaja, maka is going to produce, okay, one zygote with chromosome XXY, which is it will lead to Klinefelter syndrome. Dan satu lagi dia akan produce XO, which will lead to Turner syndrome. Okay, so as a conclusion, okay, if non-disjunction occur during meiosis 1 of spermatogenesis, it's going to produce abnormal gamete either N+1 ataupun, okay, N-1. So kalau N+1, it has both X and Y chromosome inside the cell. Okay, when it fertilized with normal ovum with 1X, okay, it's going to produce XXY, which is for Klinefelter. Manakala for abnormal N-1 with zero chromosome number 23, okay, combined with normal ovum with 1X, so dia akan menghasilkan, ini tertinggal, XO. Okay, so XO ni adalah Turner syndrome. Okay, so apa jawapan dia masing-masing? Okay, jangan disokong akta tu. Okay, so Sakina yakin tadi jawab A. Ke tu untuk soalan lain ni? Okay, so agak-agak apa soalan, apa jawapan untuk soalan ni? Okay, so jawapan dia adalah yes, A, which is XXY dan juga XO, which is XXY ni, XXY ni dia adalah Klinefelter, okay, manakala XO adalah Turner syndrome. Okay, okay. So kalau jawapan awak bukan A tu, kemungkinan awak ada silap dekat bahagian non-disjunction lah. Adakah awak terbuat non-disjunction during meiosis 2 ke? Okay. So moving to the next question. Number 11. Okay. So a chromosomal abnormality where a segment of chromosome is transferred and attached to another chromosome is called translocation, duplication, inversion ataupun all of the above. Okay. So yang ni simple. Ini adalah tajuk mutation paling senang. We are talking about the chromosomal aberration. Okay. So basically we have 4 types of chromosomal aberration. Okay, kita ada duplication. So maksud duplication adalah a region of the chromosome, okay, the duplicate. Okay, dia jadi 2. Lepas tu dia akan double up lah kat sini. So we have 2 segment of chromosome that has been duplicate. So simply dia dipanggil sebagai duplication. Okay, and for deletion, okay, for deletion pula nama dia pun dah memang deletion. Kita tahu what happen is a segment of the chromosome has been deleted. So bila deleted, dia punya kromosom jadi pendek lah. Okay, so next kita ada translocation, di mana region of the chromosome tu dia breaking off, dia tercabut, diputus. Lepas tu dia akan rejoin balik, okay, sama ada at the same chromosome ataupun another non-homologous chromosome. So kalau kita tengok dalam contoh, okay, so this segment of this chromosome and also for the red one, okay, they have been breaking off. Lepas tu dia rejoin balik but at different chromosome. Okay, hijau attach kat merah, yang merah pula dia attach dekat hijau. Okay, so this is what we call as translocation. Okay, so untuk inversion pula literally membawa maksud inverted, terbalik. Okay, di mana this segment, okay, dia terbalik. Kalau contoh kat sini gene A bawah gene B, so dia bertukar menjadi gene B dan pula jadi bawah gene A. Okay, so itu maksud inversion. Okay, so apakah jawapan dia untuk soalan ini? Obviously adalah A, iaitu translocation. Okay, so next question, question number 12. Okay, so the frequent exposure to X-rays, okay, will cause a mutation in a woman. Okay, however, she later had a normal child because the mutation was Dia kata perempuan ni selalu sangat expose dengan X-ray. Mungkin dia jadi juru X-ray kot kan kat hospital. Tapi Alhamdulillah, luckily anak itu normal. Sebab kita tahu X-ray akan menyebabkan mutation. So adakah dia adalah sejenis spontaneous mutation, germ cell mutation, induced mutation ataupun somatic mutation. Okay. So simply, mutation memang ada dua jenis. Okay, it can happen secara spontaneous ataupun secara induced. Kalau dia berlaku secara induced, usually is because of exposed to mutagen. Macam kita tengoklah tadi, contoh dia X-ray. Okay, so what is mutagen? Mutagen is an agent that can induce changes in the DNA and cause mutation. So we have two types of mutagen, physical and chemical. So physical is any kind of rays, UV rays, X-rays, gamma rays, radiation. So all that can be the physical agent. Manakala untuk chemical agent, yang paling famous yang kita akan guna adalah colchicine. Okay. So because of the mutation, it can lead to mutation in an individual. So, tapi dia kata kat situ, mak dia mengalami kemungkinan mengalami mutation tapi anak dia normal. So, konsep dia simple. If the mutation occur in germ cell, then only it can be passed to the next generation. Maksudnya anak dia pun boleh mewarisi mutation tersebut. Tetapi if the mutation occur in the somatic cells, it cannot be passed to the next generation. So anak dia boleh jadi normal. Okay. So for this case, the answer is D. Okay. Jawapan dia adalah a somatic mutation. Apa maksud somatic? Siapa boleh jawab dekat chat? What does mean by somatic? What does mean by somatic cell? Lalu kita dengar kan orang kata somatic cell, somatic mutation. So what does mean by somatic? Anyone want to try? Okay, so thank you for all the answers. Okay, masing-masing memang jawab somatic cells D. Okay. Tak sangka malam ni ikan siakap pun join saya Okay ke line kat dalam laut tu? Okay. Harap-harap tak blub lah Okay, ikan siakap pun boleh join YouTube live malam ni. Okay, so itulah pembelajaran. Okay. Okay, so kita teruskan. Tak ada siapa nak jawab siapa somatic cell Tolong siapa yang boleh tak bagi tahu apa itu somatic cell, tolong komen. Okay, so kita teruskan dengan soalan nombor 13. So soalan nombor 13, figure 2 shows the structure of plasmid. Okay, what makes plasmid a good cloning vector? So adakah because it's able to accept foreign DNA in like Z, ataupun it's able to receive the recombinant DNA through transformation, ataupun it able to accept foreign DNA in multiple cloning site, which is MCS, okay, ataupun able to maintain the structure of foreign gene, okay. So we have to know what is plasmid first, then only we can answer this question. So what is plasmid? So plasmid basically is a small DNA, okay. It is a circular DNA, okay, and it has a few characteristic, okay. Antara karakteristik of plasmid pertama adalah it can accept foreign DNA because it has MCS. So what is MCS? MCS is the multiple cloning site. It is the site where we can find different type of restriction site, okay, different type of restriction site. Okay, untuk dia cut nanti. Okay, baru keluarlah jawapan-jawapan awak ni tadi. Okay, so good. Somatic cell tu adalah semua sel dalam badan kecuali germ cells, kecuali daripada sperm and juga egg. Okay, so that's correct. Okay. So, the next characteristic of plasmid adalah, okay, it able to replicate freely in the host cells because it has ORI. Okay, so where is ORI? We can see in the diagram. So this part is ORI. So because of ORI, it can replicate freely in the host cell. Okay, maksudnya dia tak perlu tunggu host cell to replicate, dia dah boleh replicate sendiri dulu. Okay, sorry, kita tengok MCS tadi. So MCS ni adalah yang ni Okay, so MCS is located in a gene that called like Z. Okay, like ZG ni. Okay, dekat dalam like Z tu ada MCS. Okay, MCS is multiple cloning site lah, where we can find different type of restriction site. Contohnya yang kita selalu guna, kita belajar EcoR1 dan juga SMA1. Okay. So next characteristic adalah it possess selectable marker gene which is ampicillin resistant. Okay. So ampicillin resistant adalah gene yang boleh membantu dia untuk resist dalam antibiotik. Contoh dia ampicillin. Okay, maksudnya if the bacteria has this plasmid with this gene, okay, it can survive in the antibiotic sebab dia ada resistant gene. Okay, dia boleh bertahanlah dekat dalam antibiotik. And the last one is it able to express or amplify the clone gene under suitable condition. So the best answer, what make plasmid is a good vector ataupun a good carrier, okay, adalah disebabkan able to accept foreign DNA in multiple cloning site. Okay, so bila dia able to accept foreign DNA, then only it can carry the target DNA into host cell. Okay, sebab itulah dia adalah a good vector. Okay, vector ni apa? Vector ni dia macam carrier lah. Okay, dia akan carry the target DNA into host cell. Okay, so the answer is C. Sempat pula nak berkenalan kat sini korang ni. Okay, so jawapan dia adalah C, okay, good. Okay, so next, moving to the next question, 14. So in production of human insulin, okay, why mRNA is used instead of DNA molecule? Okay, part ni kena faham kan. Siapa yang ada masalah untuk jawab soalan ni, okay, tolong fokus. Okay, so kenapa, okay, when we want to make a human insulin by using a bacteria, okay, you have to use mRNA instead of DNA. Okay, so this is the story. Okay, so what happened is in eukaryotic cell, which is us human, okay, what will happen to our mRNA after transcription adalah they are going to be modified. What does mean by modified? This mRNA will undergo a splicing process where they are going to remove introns, okay, and they will join all the exon to create mRNA. So intron tu apa? Intron tu adalah non-coding region, exon tu adalah coding region. Contoh dalam gambar ni, okay. So basically inside the DNA we have exon, we have intron. So but the problem now is we don't want intron. So after transcription, the mRNA produced still has intron in there. Okay, ada banyak juga intron. So that's why it must undergo splicing process first, remove all the intron, just take the exon. So sekarang bila kita dah ada mRNA sequence yang ada exon sahaja, barulah kita boleh proceed dengan proses translation to produce a functional protein. Okay, but the problem now is if you want to make insulin, okay, human insulin, okay, from the bacteria, there are no splicing process. Bakteria ni tak boleh nak buat RNA splicing process. So if you want to ask the bacteria to produce the insulin, you must give the bacteria cDNA, which is a DNA that only contain exon without introns. Okay, so how to make this cDNA? You have to make cDNA through reverse transcription process. Okay, so this is the process di mana, okay, you have a cDNA produced through reverse transcription by using a reverse transcriptase, okay, and then cDNA will produce a DNA fragment without introns. Then only cDNA will be inserted into plasmid to form recombinant plasmid. Then only you can proceed with the process of gene cloning. Okay, then later on you can ask the bacteria to make the insulin protein. So as you can see from the diagram, okay, so from the donor cell, okay, you have to isolate the mRNA, and then when you put reverse transcriptase, it will form another single stranded of cDNA. So basically you just reverse the process of transcription. Okay, but and after that you have to degrade the mRNA because we don't want mRNA, we need DNA. So from that single strand of the DNA, okay, you put DNA polymerase, to make another strand of cDNA to form double-stranded cDNA. So this double-stranded cDNA ni lah yang you akan masukkan dalam plasmid dan seterusnya proceed with the process of gene cloning. Okay, so agak-agak what is the answer for this one? Semua dah layu dah ke? Dah mengantuk Okay, tak ada pun jawapan ni untuk soalan nombor ni. Okay, ada. Okay, ada yang jawab D. Betul ke ni soalan 14? Okay, ada yang jawab soalan nombor 14, D. Itu je lah. Thank you Izzat Aiman. Okay, for your answer. Ehsan. Okay, Christina. Okay, mari kita tengok what is the answer. So, jawapan dia adalah yes, D. Because there is no RNA splicing in the bacteria. Agak-agaknya tadi tak ada jawapan daripada semua orang ni. Mungkin masing-masing tak faham kot soalan ni. Nak kena tunggu explanation baru boleh dapat jawapan So, harap-harap sekarang semua orang dah clear lah kenapa jawapan dia D. Okay, sebab bakteria ni dia tak boleh buat RNA splicing. So, kita kena bagi dia DNA yang sesuai lah. Barulah dia akan produce the same insulin for human. Okay. So proceed with the next question, 15. So one of the steps in gene cloning is screening. So screening is done by culturing bacteria in medium agar containing ampicillin and X-gal. Okay, so dia ada ampicillin. So kita tahu ampicillin ni dia adalah sejenis antibiotik. Okay, dan dalam X-gal ni pula, dalam X-gal ni pula kita ada sejenis bahan yang macam laktos lah. Okay. So, state the importance. So, kenapa kita kena guna ampicillin dan juga X-gal dalam medium ni? Okay, so proses screening ni pun kemungkinan ramai je yang tak nampak lagi. Okay, yang macam tak faham. Kenapa apa kaitan screening proses ni? Kenapa kena pilih warna putih instead of biru? Okay, so kena faham cerita dia. Okay, so this is the story. Okay, so you have to take note, okay, during gene cloning process, okay, we are going to insert gene of interest, okay, into the plasmid. And plasmid dia ada dua karakteristik. Yang pertama, dia ada ampicillin resistant gene, di mana ampicillin resistant gene ni will make the bacteria to resist in the it can resist in the ampicillin, which is antibiotic. So maksudnya kalau you letak bakteria tu dekat dalam antibiotik pun, okay, bakteria tu boleh hidup. Tak ada masalah sebab dia ada ampicillin resistant gene. Karakteristik yang kedua pula, dia ada lactase. So lactase ni is very important to produce beta-galactosidase, which later on will hydrolyze lactose lah into glucose and galactose. So kena faham dulu dua karakteristik penting yang ada di dalam plasmid sekarang ni. Okay. So what will happen is when you insert gene of interest into the plasmid, okay, dia akan cut dekat MCS. So MCS is located inside lactate. So what happen is the insertion of the target gene into MCS in lactate, dia akan disrupt the gene. Okay, dia akan disrupt the gene, menyebabkan gene tu tak boleh berfungsi lah dan tak boleh menghasilkan beta-galactosidase. So basically there are three possibilities that will occur during the experiment. Yang pertama adalah, kita tengok yang pertama dulu. Okay, kita buat yang ni dulu. Yang pertama adalah kemungkinan during transformation, okay, iaitu during insertion of bacteria, insertion of the recombinant plasmid ataupun foreign DNA into host cell, berlaku kegagalan di mana tiada langsung plasmid yang masuk dekat dalam host cell. So apa yang berlaku? Plasmid tu dia takkan ada ampicillin resistant, dia takkan ada lactate. So bila dia tak ada ampicillin resistant, maka dia tak boleh hidup dekat dalam dalam agar plate yang ada ampicillin. So dia takkan grow lah, so yang kita akan cancel. Kes yang kedua adalah, okay, dia bawa masuk plasmid tapi plasmid dia kosong, tak ada target DNA. Maksudnya dia ada ampicillin resistant dan lactate dia masih lagi beroperasi, masih lagi berfungsi. Okay, sebab kenapa? Lactate dia tak disrupted. So apa yang berlaku adalah, okay, because of we have lactate here, okay, hydrolysis of lactose can occur. So bila berlaku hydrolysis of lactose, maka it will change the colony into blue color. Okay, untuk kes yang ketiga pula adalah di mana, Okay, recombinant DNA berjaya dibawa masuk ke dalam host cells. Okay, so we know that if recombinant DNA if target DNA inserted into plasmid, dia akan rosakkan laczac lah. So laczac tak ada, tapi ampicillin ada, so dia boleh hidup dalam agar plate. Dan bila tak ada laczac, maka hydrolysis of lactose takkan berlaku. So bila tak ada hydrolysis of lactose, the colony will remain white. Okay, so the conclusion is why we need to choose the white colony, because inside white colony, okay, means it has recombinant DNA where we can have the target DNA. Okay, so jawapan dia adalah A, okay, to identify host cell containing the recombinant DNA. Okay, so last question from me, which is question 16. Okay, so unpaired nucleotides produced by restriction enzyme at one end of DNA fragment is known as. Yang ni kita nak kena tahu pula pasal restriction enzyme. So let's look at what is restriction enzyme. So restriction enzyme basically is enzyme, dia akan buat dua benda. Dia akan recognize either specific palindromic sequence ataupun kita panggil dia sebagai restriction site. Okay, what is palindromic ni? Palindromic ni adalah apabila bacaan 5 to 3 from one strand, okay, is same with the reading of 5 to 3 prime for another strand. Nampak bacaan dia TGAA, TTC, A, yang bawah pun sama, TGAA, TTC. Bila kita baca dalam direction yang sama. So first dia kena recognize dulu sequence macam ni, which is sama ada we can call it as palindromic sequence ataupun restriction site. Lepas tu barulah dia akan cut. Okay, then only after they recognize the specific sequence, then only dia akan cut the DNA at the specific point. Okay, so for restriction enzyme we have two types. The first one is EcoR1 and the second one is SMA1. Okay, so what you have to take note for both this restriction enzyme adalah, saya dah senaraikan kat bawah ni. Okay, pertama you must know about the sequence. Okay, the sequence for EcoR1, okay, the specific sequence that will be recognized by EcoR1 is GAATTC. Wajib, fix, fakta. Okay, manakala sequence that will be recognized by SMA1 adalah CCCGGG. Okay, next you have to look at where they are going to cut. Okay, so untuk EcoR1 dia akan cut between G dan A, manakala untuk SMA1 dia akan cut between C and G. So next what you have to know is the types of cutting. Macam mana rupa, bentuk dia bila dia memotong tu. EcoR1 dia akan potong secara staggered. Apa maksud staggered tu? Staggered tu macam ni, zigzag. Okay, manakala untuk SMA1 dia akan potong secara straight cut. Dia akan potong secara straight cut. Okay, dia akan potong straight je lah macam ni. Okay, sengit. Straight cut. Okay, dan yang terakhir adalah produk dia. After it cuts through staggered cut in EcoR1, dia akan produce sticky end. Manakala untuk SMA1 dia akan produce blunt end. So apa maksud unpaired nucleotide? So unpaired nucleotide ni adalah apabila you cut secara staggered cut, it's going to produce a sticky end where we have some bases, some nucleotide without pair. Ni adalah bases yang tak ada pairing. Yang ni adalah bases yang ada pairing. So unpaired nucleotide tu maksudnya adalah nucleotide yang tak ada pasangan lah. Okay, so of course jawapan for this question adalah sticky end. Okay, so good with all the answers given from the chat. Okay, thank you to all. Saya nak sebutkan nama seorang-seorang ni panjang, banyak. Okay, so thank you for cooperation. So this is going to be the last question from me, question 16. So that's all from me. We are going to continue with the second part with Puan Tabila Rasi. But before that, okay, you can do the pop quiz given, which is the link will be given in the chat, or you can just simply scan the QR code in the slide given. Okay, scan QR code and you can terus jawab soalan pop quiz yang ada. Okay, so that's all from me, guys. Thank you so much. See you later.
Okey, terima kasih Puan Zaiti. Okey, kita tengok pelajar punya respon begitu bertubi-tubi jawapan mereka. Okey, bagus para pelajar semua sangat bersemangat tu. Okey, so saya akan share link untuk soalan quiz. Okey, you all diberi masa 5 minit untuk menjawab soalan tersebut. Okey, boleh mulalah. Sekejap. Okey, saya dah share. Okey, rasanya semua pelajar dah jawab Google Form tadi. Okey, so kita tanpa membuang masa lagi kita menjemput Puan Tami untuk ceramah yang seterusnya untuk berbincang soalan yang seterusnya nombor 17 hingga nombor 30. Okey. So saya persilakan Puan Tami boleh teruskan.
Okey, selamat petang dan salam sejahtera kepada semua pelajar-pelajar SDS kursus biologi SB015. So cikgu harap kamu dapat stay alert, calm and focus untuk second part of our discussion of Biofortune Set 2. Okay, so cikgu akan bincang soalan 17 sampai 30. Okay, sebab Puan Zaiti tadi stop at question number 16. Alright, so teacher will continue from 17 to 30. Okay. Are you still focused? Okay, you can comment at the chat column if you're still focused. Stay focused. Boleh komen. Fokus lagi tak? Baru sampai halfway through belum habis lagi. Okay. Okay, thank you Daniel for the response. Daniel, Azmi, thank you. Okay, Patricia, thank you. Alright. Okay, so we continue with the response. Okay, good. Alia, good. Alright. Okay, so question 17. Okay, during the development of pollen grain, each microspore undergoes mitosis to produce. So what is your answer? Semua dah jawab kan soalan ni 17. So what is your answer? Okay, so this is the reproductive structure of a flowering plant. It is a flower kan? Alright. So can you name the male reproductive organ here? Which is the male reproductive organ? Okay, so answer dia A. Okay, alright. We wait for the answer. Okay, the answer is A. Okay, can you give me the male reproductive organ? What is the male reproductive organ here? Fazlina jawab A. Okay. Kirtinash also A. Alright. We wait for the answer. Stamen. Okay, Khalil bagi jawapan stamen. So stamen is the male reproductive organ. Alright. Okay, stamen is the male reproductive organ. Okay. Pollen grain dihasilkan dekat mana? Kawasan mana yang menghasilkan pollen grain? Yeah, stamen. Yes, correct. Betul. Ashraf, Afika, Kitina. Okay, correct statement. Yeah. Okay. So where does the pollen grain produced? Enter. Good. All right. Enter. Okay. So now the question says, microspore undergoes mitosis to produce what? What it produce? Okay. So many of you said the answer is A. Yeah, enter. Betul. Correct. Yeah, enter is the part that produce pollen grain. Betul. Alright. A. So jawapan dia A. A tube nucleus and a generative nucleus. So now let's see the development of pollen grain. How does the pollen grain develop? So just now you answer pollen grain is produced at the anther. Betul. Jawapan kamu semua betul. Alright. Semua betul. Thank you for the answer, Balkis. Okay, betul. Alright. So anther, bila kita buat cross section of the anther, inside the anther kita akan jumpa 4 chambers. Okay, these are the 4 chambers here. And inside the chambers ni ada kita panggil dia pollen sac. Okay, and pollen sac ni contain microsporocyte. Okay, microsporocyte is the cells inside the pollen sac. So microsporocyte is the diploid cell to Anna. Okay, so microsporocyte akan undergo meiosis untuk produce 4 haploid microspore. So the question was asking, bila microspore divide by mitosis, dia akan produce apa kan? So the answer was A. Betul kan? The answer was A and you answer correctly, A. Dan dia akan produce pollen grain and inside the pollen grain ada generative nucleus and tube nucleus. Okay, dua ni. Okay, you also can call it as generative cell and tube cell. Alright. And this pollen grain, okay, is immature pollen grain. Dia hanya akan menjadi mature pollen grain selepas generative cell divide menjadi 2 sperm cell. Alright. Okay. Okay, 18. We go to question number 18. Okay, 18. Figure 3 shows sexual reproduction in a flowering plant. Okay, double fertilization refers to. Okay, double fertilization is a unique process. Dia hanya berlaku dalam flowering plant yang kita panggil angiosperms kan, flowering plant. Alright, so the question is they're asking what happens during the double fertilization process tu? Apa yang berlaku? So what is your answer for 18? Okay. Jawapan untuk 18 adalah ya, betul. Okay, correct. 17 is A. The answer for 17 is A. Okay, we are discussing 18 now. Okay, so question 18, betul D. Okay, thank you. Umi, Adib, Ashikin, thank you. Alright, the answer is A. The answer is D, sorry. 18 is D, ya. Jawapan dia D. So what is the answer? 1 male gamete to 1 egg and 1 male gamete to polar nuclei. Okay, why we call this process as double fertilization? Sebab fertilization berlaku 2 kali sebab kita ada 2 male gametes. Alright, so we see how that occurs. Ya, betul jawapan dia. Anis, Hafiz, betul? Alright, B. Okay, so this is double fertilization. Okay, where does this double fertilization occur? Dekat mana double fertilization berlaku? Can you name the part where double fertilization occurs? Yeah, D. The answer is D, correct? Yes, betul Fazlina, correct. D is the answer. Anis, Nur Adila, Afika. Okay, D is the answer, correct. Fertilization berlaku di dalam, double fertilization berlaku di dalam ovule. Teacher want the specific answer. Ovule, try lagi. Cuba lagi. Ada jawapan lain ke? Where does the double fertilization occur? Dekat mana berlaku proses double fertilization? Ovari silap madam. Ya. So apa jawapan yang betul? Ovule dengan ovari cikgu cakap kurang tepat kan? Kurang tepat ovari. Try lagi. Cuba lagi. Where exactly double fertilization occurs? Cikgu give you a clue. What you call this? What you call this? Okay. Embryo sac. Okay, Daniel, good. Fatihah, Afika, Adib. Yeah, very good. Embryo sac. So, double fertilization occur in the embryo sac. Embryo sac is the female gametophyte. So, double fertilization just now mentioned to you ada 2 sperm cell. Satu fertilize the egg to become the zygote. Okay. And the other sperm fertilize the 2 polar nuclei to become endosperm, which is triploid. Alright, so this is double fertilization. Yes, embryo sac, very good. Balkis, Daniel, okay, good. Sina, correct. Okay, so double fertilization. Okay, this diagram explains the process. So after pollination, you know what is pollination, right? Transferring the pollen grain, transfer of pollen grain to the stigma. So once the pollen grain lands on the stigma, pollen grain akan germinate. Okay, bila dia germinate, the tube cell will produce the pollen tube. So this is the pollen tube. So pollen tube will elongate, memanjang along the style. This is style. Okay, along the style, and dalam perjalanan dia nak sampai ke ovule, the mitosis akan berlaku dan the generative cell akan divide untuk produce 2 sperm. Alright. So after reaching the ovary, the pollen tube penetrates the microfile and discharge the 2 sperm into the embryo sac. Okay, and then happens the double fertilization process where 1 sperm fertilize the egg. So take note, fertilize the egg to form a diploid zygote. Alright. Okay, the other sperm will fuse. Okay, fuse with two polar nuclei to form a triploid endosperm. Okay, endosperm is the food storing tissue of the seed. All right, okay. Okay, 19. We proceed to question 19. Okay, figure 4 shows the fertilization process. What occurs during the acrosomal reaction? All right, fertilization occurs in 4 stages. Okay, there are 4 stages of fertilization, which is capacitation, acrosomal reaction, fusion of the Okay, we go one by one. All right. So they're asking what occurs during acrosomal reaction. So the answer is A. 19A. Okay, 19A ya. Jawapan dia A. Is your answer A also? Kamu jawab A juga ke? Okay. Alright. So this is the secondary oocyte, structure of secondary oocyte. So secondary oocyte, the outer layer consists of the follicular cells, which is known as corona radiata. And then we have zona pellucida here. Zona pellucida is the jelly coat that surround the plasma membrane. Okay, and then we have the plasma membrane here. Okay, this is the plasma membrane. And then inside kita ada cytoplasm dan di dalam ni ada cortical granules. Okay, this is the cortical granules yang blue color ones ni. Okay, and this is the nucleus. Alright, so structure of the sperm. So we have sperm head and you can see here the acrosome. Okay, acrosome is the organelle that you can find at the anterior part of the head. Bahagian depan head ada acrosome. So what is the content of acrosome? Dalam acrosome ni ada apa? Yeah, the answer is A. Betul? Betul jawapan dia A. What is the content of acrosome? Fazlina, A madam, saya yakin. Ya betul. Betul-betul jawapan kamu. Semua betul A. Ada enzyme. Enzyme apa? Hydrolytic enzyme. So dalam acrosome tu ada hydrolytic enzyme. So just now I mentioned to you fertilization occurs in 4 stages. So the first one is capacitation. So capacitation occurs before fertilization where the sperm travel in the reproductive tract, the female reproductive tract nak sampai ke secondary oocyte. Okay, so bila dah sampai dekat secondary oocyte, okay, the sperm comes into contact with the zona pellucida kan, kawasan ni. Okay, lepas dia dah come into contact with the zona pellucida and then baru dia akan berlaku acrosomal reaction. Yes, hydrolytic enzyme. Yes, good. Betul, Alia. Okay, alright. So acrosomal reaction is where the acrosome release the content, the hydrolytic enzyme to digest the zona pellucida. So this is the jelly coat tadi, layer ni will be digested dan sperm boleh penetrate masuk ke dalam. And the third step is fusion of sperm head membrane and the secondary oocyte. Okay, membrane of the sperm head and the secondary oocyte. Okay, dekat sini dia akan bercantum dan depolarization will happen to the plasma membrane dan this will give you the first protection yang kita panggil fast block. Okay, to prevent polyspermy lah. Alright, so after that akan ada cortical reaction. So what happens in cortical reaction? The cortical granules yang cikgu tunjuk tadi, cortical granules will be released and this will cause the zona pellucida to harden. This is known as slow block. Untuk block polyspermy jugalah. We don't want many sperm to fertilize the ovum at one time. Tak mau. Okay. Alright. So lepas tu akan ada sperm nucleus, okay, will enter the secondary oocyte dan time tu secondary oocyte akan complete meiosis 2 dan dia sudah menjadi ovum. Okay, and then the sperm nucleus akan fuse with the ovum, nucleus of the ovum, then it will form a diploid zygote. So this is all about fertilization. Right. So we recall back, okay, what happened in fertilization. Okay, the sperm contact the egg's jelly coat, but before this, capacitation has to occur. Okay, capacitation will increase the motility of the sperm and enhance the sperm function lah. Dan dia yang enable the sperm to reach and pass through the coronary radiata. So that is capacitation. Sebelum fertilization. Okay, after that, sperm contact the egg's jelly coat. Okay, and then acrosomal reaction, where the acrosome is released and they digest the zona pellucida. Okay, what is the third one? Yeah, hydrolytic enzyme. Good, betul. Fusion of sperm head membrane and oocyte. Okay, and then cortical reaction. All right. So this is the stages of fertilization. Okay, 20. We proceed with 20. Question number 20. Which of the following growth patterns best represent human growth? Okay, so here the keyword is growth pattern. So bila dia sebut growth pattern, how many types of growth pattern that you have learned? Can you remember the growth pattern? 20D, yeah. Okay. Okay, how many growth pattern? We wait for the answer. How many growth pattern that you have learned? The options here are given: intermittent growth curve, unlimited growth curve, sigmoid growth curve and limited growth curve. Kita nak cancel out the answer that is not growth pattern. Okay, ada yang jawab 6, Nik Nurin 6, okay, Balkis 4, ya, 20D, alright. So can you give the bilangan growth pattern? Berapa yang kita belajar? 6 growth pattern. 6 or 5? 5. Amirul betul, 5. Hilman, 5. Okay. Siti Nur Balkis, 5. So we have 5 types of growth pattern that you have learned. Alright, so which is not a growth pattern here? Sigmoid growth curve. So this is not a growth pattern. We cancel out C. So what is the answer? Answer dia adalah D. Okay, answer dia D, betul. Okay, yeah. Fazlina, correct. D, limited. Okay, alright. So what is limited growth curve? Okay, limited growth curve. Okay, maksud dia organism grow sampai satu maksimum steady state, dia grow, lepas tu dia stop growing. Okay, lepas dia stop growing, aging proses akan occur, proses penuaan, dan lepas tu dia akan matilah. Alright, so kalau plant daripada dia germinate sampai dia boleh disperse the seed, lepas tu dia mati. Tu kita panggil limited growth curve. Okay, kalau kita tengok yang ini adalah annual plant, contoh annual plant. Okay, annual plant adalah plant yang complete dia punya life cycle in one growing season sahaja. Okay, so kita tengok graph limited growth curve ni. Okay, kenapa ada decrease in weight? And usually limited growth ni can be represented in a single sigmoid curve. Alright, okay. So kenapa ada decrease in weight? Because the food that is stored in the cotyledon of the seed has been used. Alright. So lepas dia dah keluarkan leaf, daun, lepas tu baru dia akan buat fotosintesis kan. And that will increase the mass lah, berat pokok tu. Dan dia akan keep on increasing. And then bila dia reach penuaan, dia akan start to disperse dia punya seed. Tempoh hayat dia hampir tamat, so dia disperse the seed, lepas tu dia mati. Okay, so this is limited growth curve. Okay. Ya, betul jawapan yang kamu bagi tu 5. Kita belajar 5 jenis growth pattern. Alright. So this is the second type of growth pattern, which is unlimited growth curve. Okay, kalau unlimited growth curve, beza dia organism yang menjadi contoh untuk unlimited growth curve ni, dia akan grow sepanjang tempoh hayat dia hidup. Dia akan keep on growing. Okay, so kalau kita tengok dekat sini, contoh organism yang belongs to unlimited growth ni adalah woody perennial plant. So plant ni akan grow throughout all seasons. So kalau kita tengok graph dia, dia adalah consist of series of small sigmoid curve. Okay, which is different from the limited growth tadi kan. Kalau limited growth, dia represented by single sigmoid curve saja. So yang ini ada series of sigmoid curve lah. Because dia non-stop, dia grow sampai dia hidup. So dia hanya akan mati due to environmental factor macam natural disaster ke ataupun kita tebang dia. Okay, alright. So contoh woody perennial plant boleh jadi pokok pine, okay, pokok cengal, meranti, jati, semua tu adalah contoh woody perennial plant. Okay. So this is another growth pattern that you have learned, intermittent growth pattern. Okay, so intermittent growth pattern ni shown by arthropods, insects. So arthropods ni dia mempunyai exoskeleton. Exoskeleton adalah kulit luar dia yang keras lah, hard. Okay, so graph ni unik, macam bentuk tangga kan, dia punya graph. Okay, so intermittent growth curve ni, okay, intermittent growth curve ni dia only show growth during the ecdysis, dekat sini saja. Okay, growth only occurs during the ecdysis. Okay, so can you see here, this is grasshopper. So grasshopper dia buat ecdysis. Okay, dia remove dia punya old exoskeleton, replace dengan yang baru. So during the ecdysis process tu lah dia boleh increase in size. Okay, while the new exoskeleton is still soft, so dia boleh increase dia punya size. So only if you notice here, the growth only occurs during the ecdysis process here. Okay, so ecdysis ni also known as molting. Okay, sampai dia jadi adult. Sebelum dia jadi adult, kita panggil dia instar stage. Alright. Okay. Fazlina, okay, good. Okay, alright. Okay. Okay, isometric growth pattern. Okay. Isometric growth. So iso means you know, same. Apa yang same? All the parts of the organ, okay, grow at the same rate. So kita nampak dekat sini, change in size of the organism is not accompanied by change in the shape of the organism. Okay, to simplify this, we can say we can see increase in size, but the shape remain the same. So you can see in the fish here. So fish ni dia follow isometric growth. Okay. Okay, allometric growth. Okay, kita boleh tengok dekat sini, allometric growth. Okay. So in human, we can note the changes in the proportion of body parts from birth until maturity. So the head, size of the head is slightly bigger compared to rest of the body when we are small. The newborn, okay, size kepala dia lebih besar. As we grow, our head become smaller compared to rest of our body. Betul kan? Yeah. Okay. So growth occurs when an organ grow at different rate from rest of the body. So change in size is accompanied by change in shape of the organism. So as the size increase, shape also change. So this is allometric growth. So betul jawapan kamu tadi, ada 5 jenis growth pattern yang kita belajar. Limited growth, unlimited growth, isometric, allometric and intermittent. So 5 lah. Okay. Okay, question number 21. Okay, question ini 21 to 30 dia ada more than one possible answer. So kita panggil ni aneka gabungan. Okay, soalan macam ni. So you have to be very careful when you are choosing the answer. So usually kita akan cross out yang salah dululah, okay, untuk make our choice easier. Okay, so select the correct statement about the properties of water. properties of water kita belajar dalam topik 1, molecules of life. Okay, so I hope you still remember. Okay, so dia bagi dekat kita, so you have to cancel out the wrong one first kan. Dia bagi correct statement, dia suruh kita pilih yang betul sahaja berkenaan dengan properties of water lah. So high surface tension, betul. Yang number 2 ni betul tak? High latent heat of condensation. Macam salah kan? Okay. Salah. Sepatutnya apa dia? High latent heat of. What is the answer? High latent heat of. What word you can replace with condensation there? Sebab condensation tu salah kan? Ya. Ikan siakap nombor 2 salah. Ya, betul. High latent heat of vaporization. Yes, betul, betul. Dania. Okay. Betul, betul. Vaporization. Okay, correct. Okay, vaporization. Betul. Yes, thank you. Ramla Aisyah, okay. Alisa, correct. Nur Atira, vaporization. Yeah, correct. So what is the answer? If we cancel out number 2 salah kan? Okay. So the answer is 1 and 3. So 1 and 3 will be B lah. Okay, so the answer is B. Okay, do you remember how many properties of water that you have learned? Ada berapa properties of water yang kita belajar? Yes, good, vaporization, and the answer is B. Ihsan, betul Ihsan. Atira, correct. Ramla Aisyah, Kristina. Ya, Kristina, betul, 1 and 3. Nik Nur Alia. Yes, thank you. Akmal. Fazlina, yes, correct, B. Yeah, B. Okay, do you remember how many properties of water that we have learned? Ada 6, yes, correct. Hafiz yang selalu first jawab. Okay, 6, 6. Kita belajar 6 properties of water if you still remember. So here, 3 of the properties are given. Yes, correct Amirul. Correct. Dania, correct. Okay. So most of the properties is due to, yes Nadira, betul, 6. Okay, most of the properties of water molecule ni adalah due to the hydrogen bond. So hydrogen bond is between the hydrogen atom and oxygen atom lah. Of course you know this kan? Yes. Okay. Okay, high specific heat capacity. Okay, this is one of the properties given in the answer options tadi. Okay, so high specific heat capacity ni maksud dia high amount of heat energy that must be absorbed. Okay, absorb of energy ni untuk break the hydrogen bond. Okay, kalau loss of energy ni untuk form the bond lah, hydrogen bond. Okay, so for 1 gram of water to change its temperature by 1 degree Celsius. So from this statement, we come to know that amount of heat energy perlu diabsorb dengan banyak kalau kita nak ubah temperature of the water. So it's not easy to change the temperature of water because the hydrogen bond kalau dia nak break, dia mesti absorb a lot of heat energy. And apa importance dia? Okay, why water has this property? And apa kepentingan dia? Okay, number 1, dia akan minimize the fluctuation in temperature. Even in our body, body cells, temperature kita tak boleh berubah according to the surrounding, bahaya. Okay, sebab dalam badan kita ada enzyme dan metabolism going on, so tak boleh. And it is also important to allow aquatic environment, animals at the aquatic environment to have a stable temperature. So temperature of the aquatic environment doesn't change according to the surrounding temperature lah. Okay. Alright. So high surface tension, yes betul, 6. Natasha, correct, 6. Okay. So high surface tension is another property. This is also due to the hydrogen bond. So hydrogen bond makes the water molecules stick to each other. So process of water molecules sticking to each other tu kita panggil cohesion. Okay. So this makes high surface tension. Okay. So the importance of surface tension, this allows small organism to move on the surface of water without breaking the hydrogen bond. Sebab dia ringan kan? Okay. High latent heat of vaporization. So this was the wrong one tadi. So sepatutnya high latent heat of vaporization lah. Okay. So high amount of heat energy required to be absorbed. Absorb ni cikgu bagi tahu tadi to break the hydrogen bond kan. This one pentingnya adalah kita nak the water change from liquid state into water vapor. So daripada liquid state dia nak tukar jadi gas. So also need to absorb a lot of heat energy. So bila water absorb a lot of heat energy baru dia boleh vibrate dan water molecules tu akan bergerak dengan cepat kan. Baru dia boleh break the hydrogen bond and escape as vapor lah. Okay. So yang ini apa importance dia? Okay, importance dia adalah the evaporation of sweat on our skin and also transpiration from the leaf. Ni bila kita temperature increase, kita rasa panas and we sweat. So bila sweat, as the heat is removed, okay, bila ada evaporation of the sweat from our skin, and this will give a cooling effect, okay, to the body. So this is the importance of high latent heat of vaporization. Ya, betul. Okay, thank you for all your response. B ya, jawapan dia. Okay. Yes, Amirul, betul. Thank you. Okay, number 22. Which of the following can pass directly through the plasma membrane? Okay, kalau pass directly, okay, pass directly ni maksud dia referring to simple diffusion. Agree? Simple diffusion kan? Okay, bila kita baca soalan ni, pass directly through the plasma membrane. So, it has to be simple diffusion lah. Okay, so kalau simple diffusion, it only allows small non-charged ataupun hydrophobic molecule saja yang boleh simply cross the plasma membrane. Okay. So, kita tengok dekat sini, molekul apa yang boleh pass directly through the plasma membrane. So, gases, boleh? Ya, agree. Thank you Nick Nurin, Alia. Okay. Gases boleh, hydrophobic molecules boleh, and small polar molecules boleh. So we look at the answer options here. So we have lipid, water and glucose. Cikgu bagi tahu tadi, the way to answer this type of question is you cross out jawapan yang salah kan? Pilihan jawapan yang salah. So kita boleh tengok daripada senarai jawapan dekat sini. Okay, glucose. Glucose ni boleh tak dia pass through the plasma membrane through simple diffusion? Macam tak boleh kan? Why cannot? Why glucose cannot? Why glucose cannot? Yeah, the answer is A. But teacher asking why the glucose cannot simply pass through the plasma membrane. Yeah, A. Kirtina A. Fazlina A. Ehsan, Rizwan A. Yeah, betul. The answer is A. Correct. The answer is A. Why glucose cannot pass through? Because glucose has bigger molecular shape. Okay, Nick Nurin, thank you for the answer. Afika, it is too big to pass through. Glucose is big molecule. Glucose is polar molecule. Very good. So Alia, your both answers are correct. Okay, glucose is a big molecule, large molecule, and it is polar. So dia tak boleh simply diffuse across the plasma membrane. How about lipid? Okay, lipid adalah hydrophobic molecule. So of course dia boleh cross lah. So water ni even though they adalah polar molecule tapi they are small. So dia boleh cross the phospholipid bilayer lah. Okay. So how glucose pass through the plasma membrane? Ya, betul Akmal. Large polar molecule, correct, betul. Ya, glucose molecule are too big to pass through the plasma membrane. Yes, correct. Sebab glucose adalah besar. Ya, betul Fazlina, besar. So macam mana glucose cross the plasma membrane? Through facilitated diffusion. Okay, so what is facilitated diffusion? Facilitated diffusion from the word, kita faham, ya betul. Ya, facilitated diffusion, Hafiz, correct. Hazif, correct. Okay, facilitated diffusion di sini bermaksud ada help from the protein. Protein membantu glukos untuk cross the plasma membrane. But it is still under passive transport. Dia berlaku daripada higher concentration gradient to lower concentration gradient. Cuma with the help of carrier protein. Okay, glukos boleh cross the plasma membrane. Yes, facilitated diffusion. Hasif, betul. Okay, 23. Okay, we move on to 23. Okay, which of the following statement outline the difference between cytokinesis in animal and plant cell? Difference, kita nak tengok perbezaan dalam cytokinesis. Okay, so what is cytokinesis? Cytokinesis is division of cytoplasm dan dia berlaku selepas nucleus divide. Baru cytoplasm akan divide kan? So kita nak tengok perbezaan dalam animal dengan plant cell. So what is the answer for 23? 23, answer dia A. Okay, the answer is A, 1 and 2. So as I said just now, you have to cancel out the wrong answer kan? Kita cancel the wrong one. So animal cell have centriole and ester, whereas plant cell do not. Tadi pun Puan Zaiti ada present, discuss dalam soalan dia bagi tahu centriole, about ester, semua dia tunjuk tadi kan? Okay. Then animal cell have cleavage furrow. This is the main point. Perbezaan yang paling ketara antara dua animal cell dengan plant cell. Animal cell ada formation of cleavage furrow. Plant cell ada formation of cell plate. Okay. So the third one, okay, at the end of the cell division, the daughter cell for both plant and animal cell will develop their cell wall. Betul ke salah? Macam salah kan? Sebab apa? Animal cell mana ada cell wall? Animal cell takkan develop cell wall lah. So wrong. So number 3 we cancel out. So left 1 and 2. So jawapan yang betul adalah A. Okay, 1 and 2. So we can see here cleavage furrow. Formation of cleavage furrow in animal cell. Macam bentuk 8 ni kan. Okay, dia ke dalam. Plasma membrane ke dalam. Okay, this is cleavage furrow. And ada formation of cell plate in plant cell. Ya, A. Izzat betul. Okay. Centrioles. Okay, cikgu review balik lah. Okay, centriole. Tadi Puan Zaiti dah tunjuk dah. Okay, centriole. Function dia help to organize assembly of microtubules during cell division. So centriole ni terletak di dalam centrosome. Okay, this is centrosome and inside there we have pair of centriole. Okay. So what is ester? Ester adalah the radiating microtubule yang radiate macam ni, macam bentuk star ni. Okay, bila spindle fiber or microtubule start to form, dia akan radiate macam bentuk star. So yang tu kita panggil ester. So centriole and centrosome ni hanya wujud dalam animal cell sahaja. Okay. Cytokinesis in animal cell, cikgu dah explain tadi dah. Okay, formation of cleavage furrow. So cleavage furrow, a contractile ring forms on the cytoplasmic side of the cleavage furrow yang ini. So bila dia deepen ke dalam, further deepen inside dan dia akan pinch off into two daughter cell. So this is cytokinesis in animal cell. Okay, cytokinesis in plant cell ada formation of cell plate. Okay, cell plate ni wujud daripada Golgi apparatus. Golgi apparatus produce vesicle that contain the cell wall material. Lepas tu dia akan move to the middle of the cell and the vesicle join to form the cell plate. And this is the cell plate forming in the middle. Okay, lepas tu cell plate akan fuse with the plasma membrane and the new cell wall between the membrane will be formed and it will become eventually two daughter cells. Okay. 24. Okay, question number 24. Okay, after test cross is performed, two genes were mapped. So this is genetic mapping. Okay, genetic mapping. Okay, so kita dapat 17.2. cm ni stands for centimorgan. Okay, so 82.8% of the offspring are showing the parental phenotype. A single crossing over has occurred between the two genes. Okay, macam ni. Kalau single crossing over, when the homologous berlaku crossing over between the non-sister chromatids, okay, dia akan produce 4 possible combination of gametes macam ni ya, 4. Okay. Alright. So jawapan yang ini adalah A. A is the answer. Okay, bermaksud yang mana salah? Kalau A, yang salah adalah yang ini, number 3 ni wrong. Okay, why? Because 172 of the offspring are showing recombinant phenotype. Okay, daripada maklumat yang dia bagi dekat kita, dia only bagi 17.2 cm apart kan? So ini adalah the crossover value. Daripada crossover value yang dia bagi, kita tak boleh nak estimate how many offspring are recombinant. Tak boleh ya. So that's why we cancel this out. Number 3 wrong. So 1 dengan 2 betul. Okay. Okay, how to get this answer 82.8 ni adalah, okay, kalau 17.2 adalah the peratus recombinant phenotype, so kita tolaklah 100. 100 minus 82.8, so kita dapatlah 17.2. So 17.2 ini adalah showing percentage of the recombinant phenotype. Okay, okay. So genetic mapping is a method to determine the position of a sequence of the genes along the chromosome. So the distance between 2 genes reflect the crossover value. Kalau 2 gene ni jarak dia on the chromosome kalau terletak jauh, maksud dia chances of crossing over will occur is higher. Okay. This is the formula that we use. Okay, crossover value, number of recombinant bahagi total number of offspring times with 100. Okay, so 1% of crossover value is equivalent to 1 map unit. So 1% of crossover value ni sama dengan 1 map unit. So jarak antara 2 gene ni kita tulis dia sebagai 1 map unit ataupun centimorgan, cm C kecil, M besar, okay, centimorgan. Okay, so test cross involving link gene with crossover. Mesti ada data dia baru kita boleh calculate kita punya crossover value. Okay, cikgu dah tunjuk dah formula dia tadi. So kita akan dapat 4 different phenotype from the test cross involving link gene. Okay, with crossover. So 2 number yang kecil macam Puan Zaiti bagi tahu tadi, the 2 smaller group will always represent the recombinant phenotype. Dan 2 number yang besar dia adalah the parental phenotype lah. So bila kita letak number of recombinant over total number darab 100, kita dapatlah 17% as the crossover value, and this is 17 map unit. Maksud dia jarak antara 2 gene on the chromosome is 17 map units, 17 cm. Okay. So dengan adanya crossover value, kita boleh menentukan kedudukan of the 2 genes on the chromosome. Okay, we proceed to the next one, 25. So according to the Hardy-Weinberg law, ni topik 5, population genetic. Okay, Hardy-Weinberg law, choose a condition when genetic equilibrium is achieved. Okay, so according to Hardy-Weinberg law, he states that the frequency of allele and genotype in a population's gene pool remain constant over generation. Okay, what is the answer? C, betul. Ya, betul. Ihsan betul, C. Okay, kalau C maksud dia, yang mana salah? 1 ni salah kan? That is migration of gene. Okay, why number 1 is wrong? Random mating and no mutation betul. Okay, we look at the 5 assumption here. Okay, these are the 5 assumptions of Hardy-Weinberg. No selection, no mutation, no migration, large population and random mating. Ya, betul C. So these are the 5 assumptions of Hardy-Weinberg. So the option given is there is migration of gene. So sepatutnya no migration. No migration. Kalau berlaku migration, migration ni maksud dia ada yang pindah keluar ataupun pindah masuk ke dalam population tersebut. So kalau ada migration akan berlaku gene flow. Kalau ada gene flow dia akan ubah kita punya gene pool. So according to Hardy-Weinberg, gene pool tak boleh berubah, dia kena kekal sama kan from one generation to another. Gene pool has to be constant. Okay. Alright. So what is random mating? Another option given here is random mating kan? Okay, random mating maksud dia all the individuals in the population have equal chance untuk mate dengan another individual of the population lah. Maksud dia, dia tak pilih lah pasangan dia. Okay, dia boleh, dia tak ada kriteria ataupun requirement tertentu. Dia boleh mate dengan siapa-siapa. That is called random mating. Okay, but actually in the nature, dia tak follow pun all the assumption ni. We just assume benda ni semua. Okay, untuk kita nak nak cakap dia punya population tu at equilibrium. Baru kita boleh apply the formula to calculate the gene frequency, the allele frequency. Okay, alright. So 26. Okay, ini dah masuk topik mutation. Frameshift mutation can be caused by. So you have to choose the answer which causes the frameshift mutation lah. Okay. So answer dia A. The answer is A. So 1 and 2 betul. Okay, 1 dengan 2 betul. So yang mana salah? Yang salah adalah number 3. Okay, number 3 is wrong. Why number 3 is wrong? Because 1 says the deletion of 1 or 2 nucleotide leading to the shift in the reading frame. And insertion, ni deletion yang ini insertion of 1 or 2 nucleotide leading to shift in the reading frame. Okay. So kita tengok insertion and deletion. Okay. So this is the normal DNA, normal mRNA. Okay, and also amino acid. This is the amino acid sequence, right? So what happened kalau ada insertion? You can see here 1C, extra C is inserted. Okay, they inserted. So what happened from the point of insertion? What happened from the point of insertion? The entire amino acid sequence changes. And this is called frameshift mutation. Inilah frameshift mutation. So from the point of mutation, semua amino acid seterusnya akan berubah. Okay, dan kesan dia lebih serius compared to substitution. Same goes with deletion. Kalau satu base is deleted, okay, A is deleted. Sini ada A kan? Sini A has been deleted. So also occurs frameshift mutation where the entire amino acid sequence changes from the point of mutation. Okay, how about substitution? Substitution takkan menyebabkan frameshift mutation sebab dia only involve change in one amino acid saja. Only one amino acid changes. Okay, and we call this effect dia boleh jadi missense mutation, silent mutation ataupun nonsense mutation. Okay, for substitution. Okay, this is frameshift mutation, another diagram showing here. So same insertion akan menyebabkan the entire amino acid sequence berubah daripada normal. Okay, alright. So different with the substitution. Kalau substitute, okay, kita ubah satu base tertukar dengan base yang satu lagi. So only it affects one amino acid sahaja yang akan berubah. Okay, berbanding dengan frameshift mutation where the entire amino acid sequence berubah. Okay, alright. 27, we proceed. Ya, betul. Amirul, betul. Ihsan, okay. Okay, what makes cDNA cloning differ from gene cloning? Alright. So, do you still remember method in gene cloning? Method in gene cloning ada 5 kan? 5 method dalam gene cloning. Okay. Isolation, cut, okay, transformation, amplification, and screening. So these are the 5 steps of gene cloning. Okay, so the question is asking difference between cDNA cloning and gene cloning. Okay, the answer is C. Kalau C, maksud dia satu ni salah kan? Using DNA ligase. So DNA ligase ni dia adalah similarity. Okay, similarity between cDNA cloning dengan gene cloning. Okay, dua-dua ada DNA ligase. Okay. So these two, DNA polymerase and reverse transcriptase. These two will be only used in the cDNA cloning. Okay, what is cDNA cloning? Cikgu Zaiti tunjuk dah tadi. Okay, teacher just go through. Okay, we need to isolate the mRNA. Okay, the process is reverse. From mRNA, kita nak produce complementary DNA kan? So, kita kena guna enzyme reverse transcriptase. Okay, the name of enzyme is reverse transcriptase and it will produce single-stranded cDNA. So daripada single-stranded cDNA to make it double-stranded cDNA, kita letak enzim kedua dan nama dia adalah DNA polymerase. So we require these two enzymes to make cDNA. Okay, lepas tu baru proses seterusnya kita akan cut, okay, and insert into the plasmid and then kita akan buat proses transformation into the host cell and then the host cell will allowed to be amplified banyak. Okay, and then akan ada screening and then we'll continue with production of insulin. So this is the difference between cDNA cloning and gene cloning method. Gene cloning dia stop sampai screening, dia tak ada production of the product yang kita nak daripada gene tersebut. Kalau cDNA cloning sampai dia menghasilkan produk lah. So usually for cDNA cloning kita akan isolate the mRNA, not the DNA. Okay, the reason Puan Zaiti bagi tahu tadi sebab mRNA is free from introns. Okay, 28. Which are true of restriction enzyme? Restriction enzyme, you know, function dia adalah to identify, to recognize specific restriction site and cut the DNA lah. Okay. So naturally true. We have to choose which is true about restriction enzyme. So naturally found in some bacteria, betul. Bacteria are the one yang produce the restriction enzyme. Untuk kegunaan dia lah, tapi kita ambil, kita pakai dalam gene cloning method. Okay. Join two DNA fragments. No kan, this is not done by restriction enzyme. This is done by DNA ligase. Okay, cut the DNA at specific restriction site, betul. Okay. So the answer is B. Okay, jawapan dia B. Alright. Okay. Okay. So restriction enzyme, kita ada dua jenis restriction enzyme yang kita belajar. EcoR1 and also SMA1. Kalau EcoR1, dia punya cutting site yang dia boleh recognize adalah GAA-TTC and it will produce sticky end. Okay, this is called sticky end. Okay. The single-stranded ends, these are known as the sticky ends. Okay. Okay, kalau SMA1, dia punya cutting site yang dia boleh recognize adalah CCC-GGG. This sequence you have to remember, kena ingat nah. EcoR1 pun tadi kena ingat GAA-TTC, yang ni pun kena ingat CCC-GGG. Right? So this will produce a blunt end because it is making a straight cut. Bila dia cut straight, dia akan produce blunt end. Kalau dia make staggered cut macam ni, okay, this is called staggered cut, dan dia akan produce sticky end. Okay. 29. Choose the function of placenta, okay, from the given statement. Okay. So function of placenta, okay, placenta is the organ that develop during pregnancy. Okay, placenta they attach to the uterus, the endometrium wall they attach. Okay, so function of placenta ada three important function. Dia supply nutrient and oxygen to the baby, from mother to the baby, and also eliminate the waste product from the baby and also carbon dioxide. Dan satu lagi function dia adalah dia juga secrete hormone. So these are the three main function of placenta. So now you have to choose from the options given here. Secrete hormone to maintain pregnancy, betul. Okay. Provide oxygen for the fetus, also correct. You have to take note, bila kita sebut provide oxygen, that doesn't mean placenta is the source. Dia tak produce oxygen. Okay, dia hanya menjadi medium untuk transfer the oxygen from mother's blood to the baby. Dia transfer saja. Through apa dia transfer? Through the umbilical cord. This is the umbilical cord. Umbilical cord ni dia connect the placenta to the baby. Okay, so we can see here, this is a maternal blood vessel. Okay, dia bersambung dekat umbilical cord. Dan barulah baby boleh dapat nutrient and oxygen to the baby, and the waste product also will be eliminated. So placenta ni dia filter from the mother's blood, dia filter, dia bagi nutrient and oxygen to the baby and also eliminate the waste product. Okay, alright. So answer dia adalah D. Okay, D means semua betul. Kan, all the options here are correct. 1, 2, 3, semua betul. D, yeah. Nurul Afika, betul, ya betul. Yes, correct. D, the answer is D. Okay, hormones. Okay, what are the hormones secreted by placenta? Progesterone, estrogen, and there's one more hormone secreted by placenta. What is the name of the hormone? Ya, D, betul. Christina, betul. 1, 2, 3. Correct, all correct. What is name of the hormone? One more hormone secreted by placenta. Progesterone is there, Afika. Progesterone is here. One more hormone. Progesterone, yeah. Yeah. Cikgu dah bagi 2 dah. Progesterone, estrogen, one more. Towards the end of pregnancy. Oxytocin, no. Oxytocin is secreted by fetus and the mother's posterior pituitary gland. Bukan placenta. Estrogen dah ada dah. Estrogen, progesterone dah ada. One more hormone. Secreted by placenta, the clue is towards the end of pregnancy. GnRH, no. Nick Nurin Alia, no. Try lagi, towards the end of pregnancy, during parturition, during the birth process. Prolactin, no. Prostaglandin, baru cikgu jumpa. Afika, correct, prostaglandin. Okay, so prostaglandin enhances the contraction of the uterus, betul? So that is another hormone secreted by placenta. So now you know the function of placenta. Yes, Nurul Arina, betul. Sharifah, betul. Prostaglandin, correct. Okay. Okay, we have reached to the last question. Last one. Okay, you have to choose the correct pathway sequence of sperm movement in the male reproductive system. So movement of sperm, maksud dia kena bergerak dari tempat dia dihasilkan. So di manakah sperm dihasilkan? Where is the sperm produced? Di dalam testis, in the seminiferous tubule. So this is the seminiferous tubule of the testis. Okay. Okay, so from the seminiferous tubule of the testis, dia akan pergi dekat epididymis, kawasan ni, tubule ni, the coil tubules here, located on the top of testis. And then dia akan move through the vas deferens. Yeah, epididymis dulu and then vas deferens. And then baru dia akan sampai ke urethra. So laluan dia adalah, okay, the answer is C. One tak ada, uterus tak ada. Masa bila dia sampai ke uterus? No, okay. So epididymis and vas deferens. So the answer is C. Okay, alright. So with that, we have come to the end of our discussion. So thank you for all your response throughout cikgu punya presentation tadi. Okay, yeah, C, betul. Izzati, betul, correct. Okay. Okay, so believe you can and you are halfway there. Sikit lagi dah sampai ke hujung semester 1. So sikit lagi untuk kamu punya final exam. So do well in your exam. Okay, don't stop. Okay, when you are tired, stop when you are done. So you are not done yet. Okay, so continue. Teruskan. Okay, nothing is impossible. The word itself says I'm possible. So it is not impossible. So cikgu rasa semua boleh buat yang terbaik untuk kamu punya exam PSPM. Okay, So good luck with your exam. Okay, it always seems impossible until it is done. So do your best. So teacher wish you all the best, and I stop here and I will pass over to our Puan moderator, Puan Hidayatul. Thank you.
Okey, terima kasih Puan Tami. Okey, perbincangan penuh meyakinkan dan memang terbaik ya. Okey, okey baiklah. Saya akan share link yang kedua, link soalan yang kedua. Okey, mohon para pelajar menjawablah soalan, ada 5 soalan ni. Okey, so saya akan share. Okey. Okey, tu link yang saya dah share dah. Okey, semua pelajar boleh menjawablah. Okey, ada 5 soalan dalam Google Form tu. Okey. Tadi saya ada share link untuk borang penilaian program. Mohon para pelajar mengisi borang tersebut. Okey, boleh jawab dulu soalan tu. Nanti akan, kalau berjaya, you all akan dapat hadiah lumayan lah. Okay. Okey, okey, tadi ada masalah line sikit ni. Okey, untuk sebelum kita bersurat, saya menjemputlah Puan Tami, Madam Zaiti ya. Okey, untuk memberi kata-kata semangatlah untuk pelajar. Okey, menjelang PSPM ni. Okey, Puan Tami boleh bagi ucapan kepada pelajar.
Okay, cikgu ucapkan all the best for your PSPM minggu depan. Okay, remember quote tadi yang cikgu tunjuk, remember nothing is impossible. Okay, you can do it. Okay, I'm possible and A for bio. Okay, hashtag A for bio.
Terbaik. Okey, good luck. All the best semua pelajar. Okey, buat yang terbaik. Okey, Puan Zaiti ada ucapan untuk pelajar?
Boleh dengar saya? So
Boleh.
all the best to all the students. So insyaAllah buat yang terbaik dan don't stress lah. Okay.
Okay. Okay, all the best semua. Okay, bye. Assalamualaikum. Okay, thank you.